Okay, we've talked about 4 out of the 5 special senses, and now we're talking about vision. Vision is our photoreceptor sense. Uh, everything vision is usually referred to as optic, like optometry. And where dogs were probably the kings of the olfactory sense, humans are probably the kings of the visual sense. Our visual sense is really highly adapted in humans, and we are very fond of it. I don't know if you've noticed, but whenever scientists develop some kind of a test for, I don't know, whether someone's pregnant or whatever test we've got, generally the results are something that you can see. Like we look for a change in color in the solution, right? If we were dogs, we wouldn't have done it that way, but we're humans and we really love vision. Now, vision is pretty complicated because the photoreceptor cells that allow you to sense light, they have got pigment molecules inside of them. And the pigment molecules that are in those photoreceptor cells are different ones. Specifically, you need to know that there are 2 basic kinds of photoreceptor cells in humans. One kind is known as rods, and rods contain a pigment that's called rhodopsin. I'm pretty sure that whoever discovered it wanted to name it rhodopsin, like they found it in the rods and it has something to do with vision. But someone said, oh no, put an H in there, it'll look really Greek and sciencey. So rhodopsin. Now, rods are not a human's favorite way of experiencing light. And that's for a couple of reasons. But the biggest reason is the way rods are arranged in the back of our eye. They do not allow us to get a very detailed picture. So if you've ever tried reading very fine print in very low light, you couldn't do it, right? In really low light, you can still see the paper, you can still see that there's writing on it, but you can't read it. And that's because of the way these particular photoreceptor cells called rods are arranged in the back of our eye. The good thing about rods is they are super sensitive to light. Because they're sensitive to light, even when the light is super, super dim, you can make out outlines of things. So that's why we use rods. We'll get back to that. Now, cones, ooh, humans love our cones because cones let us see in color. Humans have got 3 different kinds of cones, and the 3 different kinds of cones are actually blue and red and green. I know you probably think, no, I know color theory. It should be blue and red and yellow. Well, maybe it should be, but it's not. It's blue and red and green. And so in a very real sense, we kind of don't see the color yellow. Our brain perceives the color yellow whenever we've got a lot of signal coming from the green cones, but no signal coming from the blue cones. And then your brain will perceive, oh, that light must be yellow. Kind of cool, huh? All right. So here we have a view of the human eye. You should have learned the anatomy of the human eye in your lab, but let's go over some of the basics real quickly. First of all, we've got our sclera. The sclera is the white part of the eye. The white part of the eye, the sclera, is actually a really, really tough connective tissue. And I know it looks like it's just right here in front, but it actually goes all the way around to the back of the eye. The eyeball itself is made out of this tough connective tissue called sclera. Now, over the top of the sclera, you see these really delicate blood vessels. Those delicate blood vessels, they're not in the sclera itself. They're actually in this really delicate clear layer of tissue called the conjunctiva or conjunctiva. And the conjunctiva is on the outside of the sclera, and that's where those little blood vessels are. And when you've got really red eyes, it's going to be because these blood vessels have more blood going through them, so they're more noticeable and your eyes look red. What's next? We've got the iris. The iris is the colored part of the eye. So if your eyes are blue or brown or green, it's because the pigment in this structure, the iris, creates that color. But the iris is actually there for an important reason. You should write this down. The iris controls the amount of light that reaches the rods and cones. You know, the rods and the cones, they need to have enough light so that they can fire off their action potentials, but not too much light or they'll get damaged. It is the iris that controls that. And how? When there's too much light, the iris will contract in a way that will close down the hole in the center. And when the center is closed down, less light gets back to the rods and cones. That protects them. When there's not very much light around, the iris relaxes in a different way and it opens up the hole in the center. By the way, the hole in the center is called the pupil. The pupil is not exactly an anatomical structure. The pupil is a hole in the center of the iris. All right, from the front. The cornea. Where is the cornea? The cornea is, do you see the shiny stuff right here? That shiny stuff, the part that if you had a contact lens on, you would put the contact lens right on it. That is the cornea. And you know what's interesting to me about the cornea is those cells are all alive. The cells that make up the tissue that is your cornea, they've got a nucleus, they've got endoplasmic reticulum, they've got mitochondria, they've got DNA, they've got all that stuff, but they're clear. How are they completely clear? Actually, I don't know the answer to that. I'll have to get back to you. Where is the retina? The retina, if I went through that hole called the pupil and went all the way to the back of the eyeball, that's where I'm going to find the retina. Oh, you should write this down. The retina is where all of the photoreceptor cells of the eye, the rods and the cones, will be found. So now we're looking at the eye from the side, right? We've got a sagittal section, and so you can see up front here, you can see up front here, this is the cornea, and in a living person that would be clear. And starting here, we've got all sclera, goes all the way around the eye, and that's the white part of the eye, right? This here in blue, you can see that is the iris. I don't know why books always make the iris blue, because really humans should have brown eyes. It would be so much healthier. It is a genetic mutation that some of us don't have brown eyes. I want to make a point here that this word pupil is pointing to the opening in the center of the iris. It's pointing to the opening, right? So it looks maybe like I'm pointing to this, but no, this thing that kind of has an onion layer appearance, that is called the lens of the eye. Let's talk about the lens of the eye. Write this down. The lens is the part of the eye that focuses light. It focuses the light. I don't know if you've ever noticed, but if you're sitting outside looking at the clouds, oh, your eyes are so relaxed and everything. When your eyes are relaxed, then these muscles right here, called the ciliary muscles, they are actually relaxed, and that causes them to pull on this structure, the lens, and when the lens is being pulled, the lens flattens out, and that focuses that distant light from the clouds. It focuses it on the back of your eye, all right? And then you're like, ooh, I got to get back to my reading, and you look down and you're looking at your book, and you'll notice it doesn't take a whole second, but there is just a moment in time for your eyes to focus. Well, what's going on then? When your eyes focus, these muscles, the ciliary muscles, they have to contract. And when they contract, they narrow down the opening, sort of like the pupil does, and that releases tension on the lens. And when there's less tension on the lens, this structure, this structure, the lens gets fat, and that focuses that near vision. So the iris is responsible for controlling how much light gets back here to the retina. The retina's back here where we find the rods and cones, but the lens is what focuses the light near and far, right? What else is important? So back here in the retina, this layer that they've got depicted as yellow, that is going to be where we find the rods and the cones, which are the photoreceptor cells that give you the sensation of experiencing light. Here we've got the optic nerve. And right before the lab midterm, you learned about the optic nerve. You learned that information is going from the back of the eye, down the optic nerve, to the optic chiasm, and then leaves through the optic tracts. If you take 150 and 151, you'll learn that in more detail. Yeah, make sure you know all of these terms that are in yellow. Why do we have a blind spot? Okay, here's why we have a blind spot. Because of the optic disc. Right here, the optic disc. Let me show you about that. Oh, before I get to that, I wanted to show you this beautiful image of the cornea. So here we're looking at a gorgeous brown eye. And you can see the iris has got all this interesting texture to it. And do you see this glassy structure right here? That's the cornea. Cornea is actually very tough, but weird that it's alive, but as clear, actually more clear than glass is. A little more detail of the anterior chamber. I do get carried away, but this is just showing us that clear cornea, and then there's the pupil in the center of the iris, and there's the lens, and light has to go through all of that. Ooh, here's what your optometrist sees when she's looking through your eye. You know how they get way too close to your nose. They're like this, oh, okay. This is what they're seeing in the back right here. Let me give you some orientation. This is the optic disc. The optic disc is the beginning of the optic nerve. So just on the other side of that clear area would be just this tube of axons that are going down towards the brain, right? And you'll notice that blood vessels are entering and leaving, this is Gidget, entering and leaving the back of the eye at the same place that all the nerves are entering and leaving. Now over here, do you see this area right here? In that area, there's a small part that's only about the size of the head of a pin where humans will do most of our seeing. And hopefully your lab instructor will elaborate on that in the lab lecture. What else do we need to know? Let's go to the next slide. I'm going to start here on the next video, all right? So we will pick up here.